So, good afternoon, everyone. I hope I am audible and the screen is also visible to you guys. Okay, so we'll start with the lecture. So, I am Rishabh and I am a PhD scholar at IIT Kanpur, and we'll be starting with the second week of the NPTEL course Biochemistry. Okay, so starting with the first question for today's lecture. What is the ratio of concentrations of acetic acid and acetate required to make a buffer system of pH 5 when you are given with the pKa value of acetic acid, that is 4.76? So, what can be the correct answer?
1.7.
Okay, so we have got the first answer. Anybody else? Okay, so the correct answer is option number 4, that is 0.575. Okay, so how will we solve this question? So remember Henderson Hasselbalch equation. Okay, so it states that the pH, just wait a minute. So the pH is equal to pK plus log of proton acceptor by the proton donor. So these are the ratio of the concentrations. Okay, so if we are considering the acetic acid case, then in that case, this would be the proton acceptor and this would be the proton donor. Okay, so here it will come CO negative and CH3COOH. Okay, so now the pH is given as 5, pK value is 4.76, so log. Now if we subtract this 4.76 from 5, then it would yield 0.24. Now if we have to remove the log, then in that case it will be 0.24, 10 to the power 0.24. Okay, and we have to calculate the ratio of acetic acid to acetate. So that would be the reciprocal of this. So that means 10 to the power minus 0.24. Okay, so if we calculate the value of 10 to the power minus, That would come around 0.575. Okay, so hence the correct answer is option number 4. Okay, is the explanation clear? Hmm. Yes.
What if pH is less than 4 or 4?
Uh,
Because
Sorry,
it is
can
10 raised to the power 0.24, it's okay. Then we have to calculate negative on both sides.
yes,
Will it 10 raised to the power minus 24 or something like that.
yes. So what was your question? I didn't get. Can you please repeat?
Here you had subtracted 4.75 from the 5.
Yes.
Suppose instead of 5, the pH is 4 or less than 4, then what we will do?
Okay. So if the pH is less than 5, and you are saying, right? So
Correct.
in that case, what will happen? This would be negative. Okay. So in that case, the whole value would change. Okay. So in that case, this would be 10 to the power. This thing would come as a positive case. Okay. So in that case, the value will eventually change after calculating.
That's my question. Ratio, I think, is it cannot be negative. That's what I am asking.
Oh,
The answer will be
okay.
in positive only.
Okay. In that case, because this is we are finding the ratio of the concentration, right? So pH cannot be negative, but the ratio can be this is also the log value. Okay, so in that case, it can go into 10 to the power negative case. So that is not an issue, because in that case, if 10 to the power negative anything comes, that would eventually lead to some less number of positive value. Okay, that can be less than 1. Like here also it is less than 1, but it's positive value. So in that case, when the ratio is like that, the it is fine. We are not talking about the pH here. Okay, is it clear?
Yes, sir.
Okay. So let's move on to the next question. So here are the different titration curves of different amino acids. Three amino acids are given here. You have to match the column 1 with column 2. So column 1 is the amino acid, and column 2 is the titration curve of different amino acids. So you have to match both the things. So what can be the correct answer? Okay, we have got first answer.
Option B.
B. Okay. Option 1, option 2. Okay. So the correct answer is option number 1. Okay, now we'll see the explanation. So what is the A amino acid?
Sir, alanine.
Yes, this is alanine, because here is the R group. So this is methyl group. In case of alanine, it's the methyl group. What is the B amino acid? Okay, it's lysine. Okay, What is the C amino acid? Because it also has another COH group. Okay, so it would be aspartate. Okay, now we'll see the different titration curves of different amino acids. Okay, so because in case of alanine there is no extra COH or NH2 group. Okay, so it would have only one amino group and one carboxylic group, so it will titrate accordingly. So if we titrate alanine, so what will happen at this point? It will go up. Okay, then it will have a stable buffering action at the lowest pH. Okay, so this is for the COH group. Now it will again shoot up, and here will be a point where the pi comes. Okay, at pi is this particular peak will come, and then again it will see a saturation type of thing, and this is for the NH2 group. Okay, and then it will go up. So this is a basic titration curve for a amino acid. So this is for the alanine. So this is option number 2. Okay, now moving on to lysine. Now, in case of lysine, there is another additional amino group. Okay, so in that case, what will happen? Because there is only one carboxylic group, so this will be a plateau, then followed by the major uh, increase because of the pi. Then here comes one plateau and the other. This is because of the two amino groups. So this is the our side chain amino group, and this is the amino group of the amino acid. Okay, so this is in case of the lysine as the third option. So this is the titration curve of lysine. And in case of carboxylic acid, we have two carboxylic group. Okay, one is the R chain and one is the for the amino acid. So now it will have two plateaus at the lowest pH. Okay, so first it will go like this, then followed by this is for the pi, and this is for the amino group. Okay, so this is the typical titration curve of the aspartic acid. Okay, so hence the correct answer would be option number 1. So is the explanation clear?
Yes, sir.
Okay, so let's move on to the next question. So this is a series of questions to understand the titration curve. Okay. So you have provided with a 100 ml solution of zero point 0.1 molar glycine. So glycine is also an amino acid. What is the side chain present in glycine?
H.
H.
Yes, it's H. So this is a only amino acid that is achiral in nature. Okay. So moving back to the question, at pH 1.72, it was titrated with 2 molar NaOH solution. Given below is a diagram of the titration curve obtained. The key points in the titration are marked as 1, 2, and 3. Okay, this is point 1, 2, and this is 3. For each of the statements given below, identify the appropriate key point. So this is a series of questions related to the same information given above. So the carboxylic group is fully negatively charged. That means it's deprotonated, and the amino group is fully positively charged. That is protonated at which point? 1, 2, or 3. So that means this is the state of the carboxylic group, and this is the state of the amino group. So at which point both of these groups are present? Okay, we have got the first answer. Then anybody else? Okay, someone said A also. Okay, so yes, the correct answer is option number 2. Okay, how? We'll see. So in case of the first point, as you can see, the pH is less than pi. Okay, so at this particular pH, only because it's reaching pKa value, pKa1 value. Okay, so now here only half of the molecules have NH3 positive. Oh, sorry. Half of the molecules will have COO negative, and half of the molecules will have COOH. Okay, so that means here it is at the first pKa value. Now, at moving on to position number 2, at position number 2, the amino acid will be present at its pi. Okay, pi means isoelectric point. So in this case, the amino acid will be present in the form of their zwitterions. So zwitterions means that the net charge on the amino acid would be 0, because both the charges will be present on that, and it is glycine, so it will be H on both the sides. Okay, so at this particular point, the amino acid is present in the form of zwitterion, and it will be fully deprotonated, and this amino group will be fully protonated. Okay, so the correct answer would be option number 2 at position 2 in the graph. Okay, is it clear?
Yes, sir.
Okay, so moving on to the next question. The average net charge on the glycine is zero point 0.5 at which of the positions? Okay, we got the first answer. Okay, anybody else? Hmm. So the correct answer is option number 1. Okay, how? I'll tell you. So in this case, what will happen? This is the original state of the amino acid. Okay, now if we consider when the pH is less than the pKa value. Okay, so what will happen at this particular point, the NH3 group, sorry, NH2 group will be protonated. Okay, and this particular thing will result in plus 1 value, but at the pKa value, what will happen, half of the molecules will be having this particular group, and half of the molecules will be deprotonated. Okay, so if we consider this particular scenario, then the net charge would result in, because half of the molecules were positive and half of the molecules were having no charge, then the value will be zero point 0.5. Okay, so the correct answer would be the first case. Okay, so is the explanation fine?
Yes, sir.
Okay, and consecutively at the third position. Okay, because in that case pH would be greater than the pKa values. So half of the molecules would have a net negative charge, and half of the molecules would have no charge. So the total charge on the glycine molecules at position 3 would be minus zero point 0.5. Okay, so let's move on to the next question.
So both answers are correct because they are asking net charge, not net positive or negative charge.
No, if it would be negative charge, then it will be minus zero point 0.5.
Okay.
Okay.
Sorry, sir.
Okay. So yeah, this is actually a tactic to confuse the students. Okay, so please be aware with that. So moving on to the next question, half of the amino groups are ionized at which of the points? Okay, anybody else? Yes. So, okay. Yes, the correct answer would be option number one, not option number three. This would be one. Okay, because just now I explained that at first position, because the pH is less than the pK value, so in that case, what would happen? The half of the amino group will be ionized as NH3 plus, and half of the amino group would not be ionized. Okay, so this would be option number one. Okay, so moving on to the next question. The worst pH range for the buffering power is at what position? Okay, any more answers? Okay, so the correct answer is option number two. Okay, why? Because at second position, when the pH is equal to pi, the amino acid is present in a Zwitter ion form. Okay, so in case of a Zwitter ion, both the charges are present on the amino acid. So in that case, what would happen? The amino acid would not be able to perform the buffering action. Okay, because in case of a compound that is able to perform a buffering action, either it would donate or it would accept the H plus. But in this case, it can either donate or accept. Okay, so it can only donate the H plus but not accept it. So that's why this is the worst region of the buffering action. So is the explanation clear?
Sorry, sir, not clear.
Okay, so uh, as in case of the pi, that is the at the isoelectric point, so you can say that at second position, it's the range in which the amino acid is present in the Zwitter ion form. Okay, so at that particular point, what will happen? It has both NH3 plus and COO negative. Okay, so for any molecule to perform its buffering action, what would be the case? Either it could accept the H plus, at the same time it could donate also. Okay, so for example, if we talk about CH3COOH, right? So in that case, this is present in an equilibrium, so either it can accept directly and it can lose. Okay, but in this case, if we consider, it can donate at only once, but it cannot it is not be able to accept it right now, because the pH is not like thus allowing it to either donate or accept it. But if we call it a case like this, or like this, so in this case it will donate and it can accept. Okay, so this is the case with the first one, and this is the case with the last one. So is it fine?
What I got is pH range at the 2, it is easy to buffer for any weak acid or weak alkali.
Hmm.
For pH, for option number 1 and 3, it's difficult for weak acid and weak alkali to buffer. That's why I thought it is a, but more it will go towards the 1 or more it will go towards the 3. They will, the buffer will try to bring it towards the 2, no?
Hmm.
So that's why it is needed. Buffering power is not needed at 2.
Okay. So in that case, what will be the if you consider that case, so when the pH, so here the role is pH. Okay. So now if the pH is low or high, so in that case it will allow the amino group and the carboxylic group to basically hand over the H plus, right? But in case of the pi, when the pH is exactly same at the pi, so in that case amino acid will not be able to perform any function. Okay, so here we are talking about the buffering power of the amino acid. So what is the best buffering power of amino acid?
It is we are talking here buffering power of glycine or NaOH.
Glycine.
Oh, then it's
Okay, because the titration curve is for the glycine, right? We are using NaOH to just prove the point, basically. Okay, any more doubts? Okay, if not, then we'll move on to the next question. Which of the following statements are true with respect to the blood buffer system? So these are the options. What can be the correct answer? There can be multiple correct answers in this question. Two and four. Okay. Any more answers? Mhm, mhm. Okay. So the correct answer is option number two and three. Okay. How? Whenever CO2 concentration is increased, okay, it will get dissolved in blood. Now when it is dissolved in blood, it will form carbonic acid. Okay, it's an acid. So whenever CO2 concentration increases due to hypoventilation, the pH would decrease because an acid is formed. But when CO2 concentration is decreased due to hyperventilation, so in that case, what will happen? This acid amount goes down. So this leads to increase in the pH. Okay. So option 2, that is CO2 increases, then pH decrease, and option 3, that is CO2 decreases and pH increases due to hyperventilation. So the correct answer would be option 2 and option 3. Okay, any doubts? Okay, so let's move on to the next question. So you have to calculate the isoelectric point of alanine if the pKa1 and pK2 values are given below. So what can be the correct answer? Four, six. Okay, so this. Okay, any more answers? Okay. Okay. So first of all, how do we calculate the isoelectric point? In case of alanine, we would have only two pKa values. Okay. So this is CH3. We have NH2. So we would have pKa value of this and this. So pKa value of COH will be 2.34 and for NH2 it will be 9.7. So in this particular case, it is like easier to calculate the pi because by 2. So here only we have two pK values. Okay. So it would be 9.7 plus 2.34 by 2. So this would lead around. So the correct answer would be option number 4. Okay. So any doubts in this question?
So what if we have to calculate pH of histidine or other acidic or basic amino acids?
Yes, we'll see that in subsequent questions. Okay, if not, then we'll move on to the next question. So here we have glutamate. Okay, so now in case of glutamate, we have three charged atoms. Okay, so then we have COH. Okay, yeah, so this is glutamate basically. Now, in this case, when we have such a scenario, we choose the formula for pi remains the same. Okay, so this is a trick. Why? Because in this case, if we see, only two groups would have the pK values that are in similar range. Okay, so in that scenario, we'll choose the pK values that are nearer to each other. Okay, so in this case, the COOH values would have similar range. For example, this would be 2.19 for this pK and this would be 4.25. Okay, so we'll take these two values because these are nearer and to calculate the pi values. So in this case, it would be 4.25 plus 2.19 divided by 2. Okay, so this would come around 3.22. Okay, Now, similarly, if we have the sorry, this is a sorry. So similarly, if we have histidine or any other positively charged amino acid, so in that case, pi would be the pK value of both. This is the amino group and this is the side chain amino group. Okay, is this fine?
Yes, sir. I got it.
Okay, so let's move on to the next question. So glutamate has these three pK values. At what pH it would be positively charged? Okay, any more answers? Okay, this also. So the correct answer is option number two. Okay, how? We'll see. So this is a glutamic acid. Okay, now if this has a pK of 2.19, this is 4.25, and this is 9.27. Now, for the glutamate molecule to be positively charged, the pH must be less than the pK values or the pI. So, if we consider at this particular pK value, if the pH is less than this particular pK value, then H plus would be confined to this position. It will not lose. Okay, similar to this particular thing. But in this scenario, if the pH is less than pK value, then it would gain a hydrogen ion. Okay, so now we have to see that pH must be less than these three values. Then only it will be having a positive charge on the amino group. Okay, so in that scenario, this is the pH that is less than, although it is similar to this particular thing, but it is less than this and this. So here the amino acid will have a positive charge. Okay, is it clear?
Sir, would you please explain Dobara?
Okay, so when pH is less than the pK value, okay, of COOH, for example, so in that case it will not lose its H plus ion because it already had lot of H plus ion in the background, so it doesn't need to lose its COOH, sorry, H plus ion. But in case of NH2, because there are lot of H plus ion in the background, it can easily accept a hydrogen ion and it can convert to NH3 plus. Okay, now we'll see the scenario for the glutamic acid. So it has three pK values, two for the COH and one for the amino group. Okay, now considering this explanation, we have to choose a pH that, in that case, the H is confined with the carboxylic group, and the H plus must be confined with the amino group. Okay, so in that case, the pH would be less than or equal to 2.19. In that scenario only, the glutamic acid will have a positive charge. Okay, so here in the pH that is given below, the 2.34 value has similar range because it's less than the pK value of the side chain carboxylic acid, the main carboxylic acid, and the amino group. So this would be protonated, and this would also be protonated in that case. So is it clear now? Okay, shall we move forward? Okay, so let's move on to the next question. Okay, so here we have the pK values of various compounds given in the table below, that is acetic acid, glycine for carboxylic acid, glycine amino group, and methylamine, that is protonated. So we have to choose a correct statement from the given statements. And this is more than one option correct. So please have a look and state the correct answer. Okay, so 2, 7, and 8. Sorry, 2, 7, and 8. Okay, any more answers? Okay, So the correct answer is 2, 4, and 7, 8. Okay. So we'll see the explanation statement by statement. So for the first statement, if we see, sorry. So first we have to check what is the acidic strength. Okay, so acidic strength we generally denote based on the Ka values, that is the dissociation constant of an acid. That means how easily an acid can donate their H plus ions or the protons. Okay, so and the opposite is the pK values, because here the pK values are given. So higher the pK value, the lesser the acidic strength of an amino acid or the group that is given. Okay, so if we see from the uh, basic the pK values that are given, so the acidic strength of glycine is the highest among the groups that are mentioned. Okay, so this would be the first. Then comes the acetic acid, then comes the glycine or amino group, and then comes the methylamine protonated. Okay, so, in the order that is given below, the methylamine is the highest, then comes the glycine amino group, then acetic acid, and then carboxylic group. So, this is a false statement. Okay, now moving on to the second statement. The carboxylic group of glycine is more acidic than that of the acetic acid due to zwitterion stabilization when the former loses a proton. Yes, this is correct. As we can see from the pK values itself. So 2.34 is eventually less than the 4.76. So it is more acidic because of the zwitterion stabilization. Okay. Now moving on to the third statement, the amino group of methylamine is less basic than that of the glycine due to zwitterion stabilization when the latter gains a proton. So in this case, the glycine group is more basic than the methylamine. Okay, so this is not the correct statement. Now, the fourth option, in a solution of glycine at 2.34 pH, there is an equimolar mixture of NH3 plus CH2COH and NH3 plus CH2CO negative. So, yes, this is the correct answer. As we have seen in the titration curve also, at this particular pK value, it will have a positive zero point 0.5 group. Okay, so in that case, what will happen? Half of the molecules will be like this, and half of the molecules will be like this. So there would be no net charge in this case, and this would be positive one. So half of the molecules, sorry, total net charge on the glycine molecule will be plus zero point 0.5. Okay, and the fifth statement, in a solution of glycine at 9.60, there is an equimolar mixture of NH3 plus CH2COH and NH2CH2COH. No, this is false. At 9.60, what will happen? This would be NH2CH2COO negative and the zwitterion form, that is NH3 plus CH2COO negative. These two charged moieties would be the highest at pH 9.60. Okay, now moving to the sixth statement, the titration curve of glycine against sodium hydroxide will have a lower number of equivalence points than that of the acetic acid. No, this is also not correct. Then at point 7, at pH 2.76, acetic acid is approximately 1% ionized. Yes, this is correct. Okay, as we have seen in this case. And at point pH at 4.34, the carboxylic group of glycine is approximately 99% ionized. Yes, this is also correct. Okay, so the correct number of statements is 2, 4, 7, and 8. So anybody has any doubt? Okay, so let's move on to the okay, so let's move on to the next question. The pK values of the nitrogenous bases found in the nucleotides of DNA and or RNA are as follows. The solution of DNA is kept initially at a pH of 7 and a concentrated base is slowly added to the solution. Choose the correct statement from the options given below. And here are the pK values of different bases of the different nitrogenous bases are given. So what can be the correct answer? 2 and 3. Okay. Oh, sorry. Okay. Any more answers? 4, 1 and 6. 1, 4 and 6. Okay. Any more answers? Okay. So the correct answer is option number 2 and 3. Okay. So let's see the explanation. So we have to check the solution was at pH 7 and then a concentrated base is slowly added to the solution. In DNA, the proton most susceptible to being ionized can be found in cytosine. Okay, so there is no claim for this statement. Okay, because all the four nitrogenous bases have equal propensity to form the hydrogen bonds. Okay, so if we are moving from pH 7 to the basic pH, in that case, any of these can lose the uh, protons. Okay, because this is also less than like this is also more than 10.1 more than pH 7. This also, and this also. So all will be already protonated in that scenario. Okay, so this is wrong. Moving to the second option, when the base is first added, the pH increases, but the fraction of double stranded DNA remains close to 1. Okay, so what will happen? Although the pH increases, the double stranded DNA will be same. So the DNA will not denature at this point of time. Okay, so here what this statement is trying to say is the DNA will be in a double stranded form and it will not denature. Okay, both the strands will not separate out immediately, although the pH is increasing. Okay, now moving on to the third point. As the pH approaches 9.8, the fraction of double stranded DNA rapidly decreases. Okay, why? Because now the guanine molecule, okay, will start being ionized. That means it will start losing its proton. So in that case, the fraction of double stranded DNA now will be decreasing. So the double stranded DNA will start denaturing right now. Okay, now the base abstracts protons from the nucleobases and prevents hydrogen bonding, leading to the denaturation of the DNA. Yes, this option is also correct. Okay, because the base basically takes out the protons from the nucleobases and it prevents the hydrogen bonding between the bases, and this leads to the denaturation of the DNA. So it would also be adding in this particular answer. Okay, now if an acid was added instead of a base, the fraction of double stranded DNA would not change. Yes, because all the DNA molecule is already protonated. If we add an acid, in that case, the pH would decrease. But in that case, what would happen? It would hamper the hydrogen bonding. Okay, so the double stranded DNA would definitely start changing because the hydrogen bonding will be hampered because of the very low pH of the solution. Okay, now, the secondary structure is RNA will get denatured at a higher pH than DNA when a base is added. No, this is not a correct statement because in both the scenario, being it RNA or DNA, when we add a base, that means the pH is increasing. Okay, so in that scenario, both DNA as well as the secondary structure of RNA will get denatured. Okay, so this statement is also false. So the correct answer is option number 2, 3, and 4. So any doubts in this particular question?
Sir, could you please explain fifth and sixth point again?
Okay. So, likewise, whenever we are maintaining a double stranded DNA, it would be at nearly 7 pH. That is the physiological pH of the body. Okay. Whenever the pH goes beyond 7, that means either it increases or it decreases. In that scenario, the hydrogen bond between the bases is hampered. Okay, so hampering of this particular hydrogen bond will lead to denaturation of the DNA. Okay, so the fifth point states that if we add an acid instead of a base, the double stranded DNA would not change, the fraction would not change. So, this is a false statement because if the hydrogen bonding is hampered, that means the DNA will start denaturing at lower pH also. Okay, and at the sixth point that we were seeing, that the secondary structure of RNA will get denatured at a higher pH than DNA when we add a base. So RNA is generally a single stranded moiety, okay, but it forms a secondary structure depending upon the bases that are present there. Okay, so or either it's DNA or RNA. Okay, in both the cases, it is the hydrogen bond that is being helping the RNA to form the secondary structure and DNA to form the double stranded structure. Okay, so if we add a base, that means it is increasing the pH. Okay, now this scenario will affect both DNA and RNA, not only RNA or not only DNA. So RNA will start losing its secondary structure and DNA will start denaturing. So this is what the second point, 6 point says, that RNA secondary structure will get affected. at higher pH but not DNA. So this is not the scenario. Both the nucleic acids, either DNA or RNA, will be affected at higher pH. Okay, is it fine?
Yes, so clear.
Okay. Any more doubts? Fourth number. Okay. The base abstract proton from the nucleobases and prevents hydrogen bonding, leading to the denaturation of the DNA. So yes. So what will happen, as the initial pH was 7, right? So in that case, all the bases were protonated, leading to the hydrogen bonding between the two strands. Okay. Now, as for example, if we take adenine, so pKa of adenine is 10.1, right? If we add a base and its pH leads to 12, so now the pH is greater than pKa value, right? So now it will release its hydrogen ion. Oh, sorry. Yes. So now what will happen? It will release its hydrogen ion and it will become deprotonated. Okay, that means the base that we had extracted this H plus ion from the base and from the nucleobase, and this leads to the denaturation of the amino acid. Okay, is it fine now? Okay, shall we move on to the next question? Okay, so the next question is, lysosomes are acidic organelles in which molecules are broken down by hydrolytic enzymes. If these enzymes leaked into the cytoplasm of the cell, they could degrade many cellular components, leading to cell death. However, these enzymes have low activity in the cytosol. What might be the reason? So, what can be the correct answer from the given options? Okay, someone said fourth, first, third. Okay, any more answers? Okay, so if we consider lysosome, okay, this is a cell. There is a acidic compartment in a cell that is called as a lysosome. Okay, so here the pH is acidic, and all the enzymes that are present in this acidic pH can work only at that particular environment only. Okay, because the active site is maintained in such a way in those particular organelles. Whenever they are leaked in a cytoplasm, the cytoplasm is maintained near to 7 at pH 7. That is the physiological pH. So if they move from a acidic environment to the basic environment or neutral environment, then in that case what will happen? The enzyme's active site will not be maintained in such a scenario. Okay, so here the same concept comes, because pH is less than pK value, the active site amino acids will be protonated. Okay, so this protonation basically leads to the amino acids that are having its active site. Okay, so whenever these enzymes come in the cytoplasm at pH nearly 7, these amino acids are deprotonated. So here the amino acids will be deprotonated. Okay, leading to inactivation of those particular enzymes. So in that case, the correct answer would be option number 2, because the cytosolic pH is higher than the optimum pH for those enzymes to work. So these are inactivated in the cytosol. Okay, yes,
Cytosolic pH I think is 7.34 and lysosomal pH is 2.
yes,
So it's higher. So the optimum pH, then the optimum, so, but it can, lysosomal enzymes can cause the destruction to the organelles, not to the cytosolic pH.
no. The question is because, okay, let's see the question first. Okay, so the lysosome has these hydrolytic enzymes, right? Hydrolytic enzymes would have, like, how do they function? So basically the question is the functioning of the hydrolytic enzymes. It is at a particular pH, right? And lysosomes has a acidic pH. So at the acidic pH, the hydrolytic enzymes function well, right? Now, if these enzymes are leaked, then they could, okay, they could degrade. They could degrade. They are not degrading the organelles. They could degrade, but they are not able to degrade these cellular compartments because of their low activity in the cytosol, right? So, what is the reason of this low activity in the cytosol? That is the question. Is it fine?
So these lysosomes, they play role in apoptosis.
Yes.
So at that time, I think they are leaked in more amounts to cause damage to the cell organelles.
Yeah, so during apoptosis, many things change. Okay, so in that case, what will happen? The pH of the cell also decreases. Okay, so actually, apoptosis is a huge mechanism, and many things play into role for apoptosis. These are different cells, that is outside stimulus. Then inside also many proteins have a role, and then comes the physiological conditions like the temperature and pH. Okay, so many things are involved in apoptosis, and yes, during apoptosis the pH of the cell also decreases, such that the lysosomal enzymes also start working outside the lysosome also. Okay, any more doubts? Okay, if not, then we'll move on to the next question. So the equilibrium constants of a reaction at two different temperatures can be related by the want of equation, that is given by the following equation. And assuming the density of water does not change appreciably between 25 and 37 degrees Celsius, calculate the pH of water at 37 degrees Celsius when you have given the enthalpy change at enthalpy change of water. Okay, so what can be the correct answer? Okay, 6.81. Okay, any more answers? Hmm. Anybody else? Okay, so the correct answer is 6.81 only. Okay, now how do we calculate it? So this is the want of equation that has been given. Okay, now if we consider the second temperature to be 25 or whatever it is. Okay, so you have to take into account, if you are taking second temperature as 25, then this would be 10 to the power minus 14, because kW at 25 degrees is 10 to the power minus 14. Fine. Then, and T1 is 37. Okay, so whenever you are equating both the, like, temperatures, then it would be in Kelvin. So you have to convert degree Celsius to Kelvin. So for 25 it would be 298 Kelvin, and 37 it would be 310 Kelvin. Okay, now equating all the values, it will be, okay, so on solving this we would get the K equilibrium value at 37, and from that we'll calculate the pH of water at 37 degrees Celsius. So this would come out to be 6.81. Okay, is the explanation fine? Okay, if you guys have any problem in calculating, you let me know. We'll discuss this in the next class also. Okay, so let's move on to the next question. So consider the following peptides. When mixture of these peptides are subjected to the following chromatographic techniques. So in different experiments, we have applied these chromatographic techniques. And so which polypeptide will be eluted first, we have to figure it out, and we have to state the answer in the number of peptides that will be eluted first, as mentioned here. So what can be the correct answer? Okay. Any more answers? I think you didn't get the way of answering the question. So the question is on asking that if we are using ion exchange chromatography for the first option, for example A, which will be eluted first? So for example, if it would be 1, 2, 3, 4, so you have to answer as 1, 2, 3, and 4. Okay, so we are not asking about the order of the peptides in which this will be eluted. We are asking about the answer in which the peptide will be eluted first would be answered. Okay, so in that sense. Okay, let's discuss. So first is an exchange chromatography with polymer beads coated with positively charged functional groups. Okay, yes. So the correct answer is 4136. Okay, so in case of the first option, when the positively charged functional group is there, that means the positively charged peptide change would be attached to the polymer beads, right? So in that case, the most number of amino acids that have the positive charge are present in peptide number 4. Okay, as you can see here, lysine, arginine 2, 3, 4, 5, 6, 7. 7 positive amino acids are present. Okay, so if we have to separate using a ion exchange chromatography with positively charged functional group, we will see or we will elude the peptide that is having most number of positively charged amino acid. So that is option number 4. Okay, so in this case, fourth peptide will be eluded first. Now moving on to the B group, that is an exchange chromatography with polymer beads coated with negatively charged functional group. So in this case, the peptide with more number of negatively charged amino acid will be eluted first. Okay, so that is option number 1. In that case, 1, 2, 3, 4, 5, 6, 7, 8. 8 negatively charged amino acid are present. Okay, so in the second case, the first peptide will be eluted first. Okay, now moving on to the size exclusion chromatography. So in the given peptides, this peptide, that is the third one, will be having more size or more bulkier than size, because tryptophan moiety is maximum in this case, and tryptophan is the bulkiest amino acid. Okay, because of the indole group that is present in that as a side chain. Okay, there is another peptide, that is the peptide 5, that is also the bulkier because of the number of amino acid, but the side chain of those amino acid is small, because as I can see, there are more number of glycine residues in this peptide. Okay, so it will not be the bulkier. The bulkier would be the third peptide. So it would be eluted first in case of the size exclusion chromatography. Okay, now the last one, that is affinity chromatography with polymer beads carrying moieties that can interact with arginine. Okay, now in case of the D1, the arginine is a positively charged amino acid. So it can interact with the moieties that are negatively charged. Okay, so this means that either the peptide that have most number of the negatively charged amino acid can interact with that, or this one, because this have functional group also. For example, serine, then asparagine. Okay, glutamate and aspartate can directly interact with this. So in the last one, both sixth and first can interact. Okay, so the correct answer could be 4136 or 4131. Is the explanation fine? Okay, so let's move on to the next question. You are designing agarose beads for the use in an ion exchange chromatography column. The column is being designed to separate lysine from a mixture of amino acid, that is glycine, alanine, leucine, tryptophan, and lysine. Which of the following chemical moieties can be coated on the agarose beads to achieve this purpose? So, what can be the correct answer? More than one option can be correct in this question also. 2, 3, and 5. Okay, 2, 3, and 5. Okay, so the correct answer is 1, 4, and 6. Okay, how I can relate to? There are two things. First is the immobile phase of a chromatography column. Okay, so this is agarose beads. Now we have some counter ions, right? That can basically replace the particular compound that we have to separate from the given column. Okay, so that can exchange basically. So now what will happen if we have to separate lysine from the given mixture, then the counter ions would be positively charged. Okay, so now if we see that diethyl aminoethyl, okay, so this will have a positive charge. Then we have diethyl aminopropyl, this would also have a positive charge. Then we have quaternary amine, that is also a positively charged. So in that case, these counter ions will be attached to the agarose beads. Once lysine comes, these lysine will be replaced by these counter ions. Okay, the moiety that is attached to the agarose molecule for attaching these counterions would be negatively charged, right? So in that case, the negative ions would be present. But if we have to talk about the counter ions that will replace for lysine, so in that case, it would be positively charged compounds as mentioned here. Okay, is it fine? Okay, so what is the doubt?
Sir, I didn't understand the concept of counter ions. Whatever happens in the gross beats, if we want to separate a molecule with the negative charge, we tend to have the agarose bead with the positive charge, so that it will bind and it will be remain there in the matrix, so that we can extract in the subsequent step.
Yes.
But I didn't got like the counter ion charge should be opposite, that we are taking in the relation with the attraction or the repulsion of the separating molecule.
Okay. So what will happen? Uh, what generally we consider is this is a agarose bead. Okay, there would be some charged moieties that will be present, right? If we are isolating lysine, for example, that would be positively charged. So these moieties that are attached to the agarose beads would be negatively charged. Okay. Now, lysine would easily attract those negatively charged. So, this point, I think you have no doubt in this till here. Now, these negatively charged moieties that are present on the agarose beads are like to manage their charge, right? To make them neutral, we have some counter ions. Okay, because these moieties that we have here, right, can interact with any positively charged molecule that come in its way. Okay, so in that scenario, what will happen? The lysine that we have to separate out from the mixture will not have any space or have because of the steric hindrance. Okay, so it will not be able to bind this if other charges are bound to it. So what we do is, to maintain this, we have some counter ions that have similar structure as compared to the lysine molecule. Okay, that have been attached to this particular agarose beads. Now when the lysine comes and the pH is stable, at which the lysine is positively charged, so in that case lysine basically replace those counter ions and those counter ions go into the solution. Okay, so here we are talking about the counter ions that will be attached to these negatively charged molecules, so that if we have to separate lysine or any other positively charged moiety, can basically replace those counter ions and attach to the agarose beads. So now it's fine.
Sir, but the question is, which of the following chemical moiety can be coated on agarose bead?
Hmm.
So
Okay,
it
so
could
in
be
that
like
scenario,
two,
okay, okay, okay.
three,
Yeah,
if
yeah,
not
yeah.
the counter ion aspect.
Okay, so in that case, what will happen? Okay, considering the question, if we see, because here the counter and thing is not mentioned, so I also didn't look at it. So yes, if the chemical moieties that we have seen here, then in that case, for the negatively charged wala point, the option 2, 3, and 5 is fine. Okay, because these negatively charged beads are because of the negatively charged groups that have been added to the agarose beads. But for the counter ions wala point, these option 1, 4, and 6 would be correct. Okay, now it's fine, because here I
Understood.
didn't look at the question properly, so the counter ions were not mentioned in the question. So that's
Understood.
meant.
Understood.
Okay. So any more doubts? Okay, so let's move forward. So, in biotechnological applications, when a recombinant protein is to be expressed in a host and then purified, the plasmid containing the gene of interest is often flagged by a series of 6 to 9 codons encoding for a certain amino acid, either on the 5' end or the 3' end of the uh, gene, forming a tag. Okay, as you can see in the image also, the protein coding gene is followed by a tag. Okay, so this is 6 to 9 codons of a particular amino acid. To obtain the recombinant protein, the cell lysate is subjected to suitable treatment and the proteins of the cell are separated from the other biomolecules. The mixture of the proteins is then passed through a column containing agarose beads coated with a coordination complex of Ni2 plus and nitriloacetic acid, that is NT. Imidazole rings have an intrinsic ability to interact with this complex. This interaction is used to purify the recombinant protein and separate it from the other proteins being expressed by the host. Which of the following statements is or are true from the given statements? So, is the question clear?
Option A.
Option A. Okay, 1 and 3. Okay, any more answers? 2 and 4. Okay. So the correct answer is 1 and 3. Okay. So what happens is we generally have a his tag, that is histidine tag. So either at the 5' or at the 3' end of the, this is the 5', this is the 3' end of the gene, we have either 6 to 8 or 6 to 9 codons of histidine. Okay, so it is mentioned as 6 hashtag or 9 hashtag as the number of codons may be. Okay, so now this, if this tag is attached to the protein, then it is easier f- to be separated from a mixture of proteins using a affinity chromatography. Okay, so the agarose beads would have an antibody related to his tag, and then it will directly be attached to that particular antibody. Okay, so this have the high affinity towards the his tag itself. Right, now if we have to, for example, coated with coordination complex of Ni2 plus and NTA. So NINTA also have a uh, affinity towards the his tag. So the his tag directly attaches to the NINTA column, and while separating we use imidazole as a separation part, because histidine has a imidazole side chain. So imidazole high concentration will lead to purification of the histag proteins. Okay. Now, so the correct answer would be, because it's a affinity chromatography and we are using polyhistidine here. So the correct answer is option 1 and option 3. Okay, is it fine?
Yes, sir. Actually, I did this concept in my university in the first semester, so I am understanding this.
Okay, so let's move on to the next question. So which of the following statements are false in this case? Okay, so these are the statements related to the SGS page, and which of the following statements are false? Okay, 1, 3, 6, and 8. 1, 3, 6, and 8. Okay, any more answers? 4 and 6. Okay, any more answers? 4, 6, and 2. Okay. Okay, so the correct answer is 1, 2, 4, and 6. Okay, so let's see the statements. So first statement is chromatographic techniques are preparatory in nature, whereas electrophoretic techniques are analytic in nature. So it's the vice versa. Okay, because chromatographic techniques are very much analytical, but the electrophoretic techniques are very much preparatory in nature. Okay, so the first option is false. Moving to the second option, although DNA molecules are usually separated by page polyacrylamide gel electrophoresis, agarose gel electrophoresis is used to separate DNA oligos generated during sequencing by synthesis method. This is also vice versa. Okay, agarose gel is routinely used to separate the DNA molecules. Okay, but if we are doing DNA sequencing using DNA oligos, that is synthesis by sequencing by synthesis method, in that case, page is generally preferred because of its uh, like high sensitivity towards separation. Okay, so it can easily separate the smallest difference in the molecular weight. Okay, so second option is also false. Moving to the third option, tetramethyl ethylene diamine is used as a catalyst for the polymerization of acrylamide during gel preparation for the SDS page. Yes, this is the correct answer. So, a TEMED is actually a catalyst that leads to the polymerization of acrylamide. Okay. In agarose gel electrophoresis, gel formation is due to a chemical reaction, while in polyacrylamide gel electrophoresis, gel formation is due to hydration. No, this is also vice versa. The page gel is formed by the chemical reaction of TEMED, APS, acrylamide, and bisacrylamide. But agarose gel electrophoresis is hydration based. Okay, so this option is also false. The running buffer in agarose gel electrophoresis is tris acetate EDTA, and whereas the running buffer in SGS page is tris glycine. So yes, this is correct. Okay. The gel in SGS page is thick to prevent the aerial oxidation of acrylamide to acrylic acid. No. The to prevent the oxidation of acrylamide, we use two plates. Okay, these two plates are there, and the gel is very thin, not thick. So the thick gel is formed in agarose gel. So this option is also false. Okay. Moving on to the seventh statement. Ampholites are used to establish an electric potential gradient for isoelectric focusing. Yes. Isoelectric focusing uses PI, that is isoelectric point, to separate the proteins. So now ampholites are used to make a pH gradient. Okay, so that each protein that have that particular isoelectric point will be stopped and separated from the other proteins that are moving in an electrophoresis. Proteomic studies often make use of the 2D page. Yes, the 2D page basically involves two techniques, that is SDS page and the isoelectric focusing. So these separate on the basis of the size and on the basis of their PI. Okay, so proteomics based studies often uses 2D page as an analytical tool. In Lamelley's discontinuous page, the stacking gel has a larger pore size than the separating gel. Yes, so stacking gel has a larger pore size because they use low acrylamide, and the reason is because all the proteins that have been added to the page will be at the same point. So to prepare the protein for the separation is basically the role of the stacking gel. So this option is also correct. Okay. Now the last one, glycerol is added to protein samples to ensure they sink to the bottom of the wells created in the polyacrylamide gel. Yes. So this helps in basically increasing the weight of the protein. Okay. So the correct answer, that is the four false statements, are 1, 2, 4, and 6. Okay. So any doubts? Yes.
Sir, you are saying that electrophoresis is preparatory in nature because we need to form that agarose gel so that we can analyze the fragment or the DNA. While in the case of chromatography, we don't need any preparatory phase as such. We just need to load our samples in order to analyze the later part.
Yes, because we directly analyze the proteins, or we can separate them in using chromatography. But in case of electrophoresis, that is only in case of the preparation.
Okay, sir. Thank you.
Okay. Any more doubts?
So option 10.
Option 10. Right. Option 10, right?
Yes, sir.
Okay. So whenever we are adding any protein or DNA, okay, this is not related to protein only. Any protein or DNA molecule that we are running through the gel, either it is a agarose gel or a page. So in that case, we make a mixture that we call it as a loading dye. So that dye have bromophenol blue, glycerol, and the buffer. Okay, so bromophenol blue has a function that it basically helps in visualizing the position of the band while running the electrophoresis. Okay, glycerol helps in increasing the weight, so that whenever we are loading in a well, the for example the well is this much deep. So if we are not applying any weight to this particular sample, this would float out. Okay, so to help the sample to go deep into the well, we add glycerol. Okay, so this will help it to sink to the bottom of the gel, and now it will help in running through the gel.
Okay.
Okay, any more doubts? Okay, if not, then we'll move on to the next question. So the pI of 4 protein is given here, and if we have to separate protein D from a mixture of proteins in a single step using ion exchange chromatography, what combination of buffer pH and resin combination would you select? So what can be the correct answer? B. Okay, that means at pH 2, right? Okay, any more answers? 3. Okay. Okay, so the correct answer is option number 4. Now, the same concept applies here. When pH is less than pi, the protein is positively charged. Okay, so if we see the pi is 9, so anything that is less than 9 would yield a positive charge, and it would require a cation exchanger, right? So here the options are pH 11, so that means it is greater than 9, so it would require an anion exchanger. So the first option is wrong. The second option, pH 2, it requires a, that means it is less than 9, okay, and requires an anion exchanger. So this is also false. Now pH 6, the same reason, it requires a cation exchanger, not an anion exchanger. So option 3 is also false. Now fourth option, at pH 8, it would be positively charged, so it will require a cation exchanger to separate the protein. So option 4 is correct. Okay, any doubts? Okay, if not, then we'll move on to the next question. Three proteins in the sample were separated using SDS page. The first protein has a molecular weight of 10 kilodalton and migrated 15 centimeter. Protein B has a molecular weight of 100 kilodalton and it migrated 5 centimeter. And protein C migrated 10 centimeter. So, what is the approximate molecular weight of protein C? So, what can be the correct answer? Okay, any more answers? Okay, any more answers? Okay, so the correct answer is option number 3. Okay, so whenever we run a SGS page, we plot a graph between log molecular weight and the relative migration based on the die front that we are using. Okay, so this yield a negative slope graph. So this would be y is equal to minus m x plus c. So this is how the equation goes. Okay. Now for the simplicity of the calculation, we'll be using this as the relative migration, the values that are given here. So for the first protein, that is protein A, we have 10, sorry, log 10 that moves through 15 centimeter. Okay, because we don't know the slope and the y-intercept yet. So we have to calculate that, and accordingly we can calculate the molecular weight of protein C. Okay, and for protein B, we have log 100 minus m into, sorry, this is 15, this is 5, and C. So log 10 will be 1 and log 100 will be 2. Okay, so after we calculate this, for example, if we do this, this would cancel out and this would be 10m 1. So m is equal to zero point 0.1. Okay, and if we put m as zero point 0.1 in any of the equations, it would be minus zero point 0.1 into 15 plus c. So this would be 1.5. So c would be 2.5. And for the protein C, if we form an equation, that would be log molecular weight minus zero point 0.1 into 10 plus 2.5. So that would yield 1.5 minus 1.5. So log molecular weight is equal to minus 1.5, and we have to remove log. So molecular weight will be equal to 10 to the power minus 1.5. Now this value yields around 31.59. Okay, so this is 31.6. So option number 3 is the correct answer. Okay, is the explanation clear? Okay, let's move on to the next question. You wish to determine the molecular mass of a newly discovered protein. To do this, you run a sample of this protein on an polyacrylamide gel containing SGS. You use known proteins as molecular weight markers. The data you obtain is as follows. So we have the relative migration and the molecular weight of the protein. Determine the molecular weight of the unknown protein in kilodalton. Report your answer to the nearest integer without the unit. So what can be the correct answer? Okay. Any more answers? Okay. So the correct answer is 16. Okay. So the same concept applies here like the last question. So you will have a log molecular weight and this would be the relative migration. So you will form two equations for the first one, that is log 45,000 minus m, that have relative migration of 3.9 plus c, and the second equation, that is 14,400 log minus m into 6 plus c. So after solving this, you will get the values of m and c, and then for the other one you will put 5.8 plus c. So you'll put those values, and based on that the molecular weight would be 16. Okay, so please solve it, and if you have any problem you let me know in the next class. So let's move on to the next question. In eukaryotic cells, disulfide bonds are formed in the rough endoplasmic reticulum and the mitochondrial intermembrane space. The bonds are mostly found in secretory proteins, lysosomal proteins, and the extracellular domains of the membrane proteins. What does those tell us about the regions these proteins are present in? So, what can be the correct answer? Okay, these are reducing environments. Any more answers? Okay, so the correct answer is option number two. Okay, because the disulfide bonds are formed between two cysteine residues because it has a sulfur as a side chain. Okay, if we have to break this bond, we have to reduce this, right? So whenever in any secretory protein or any lysosomal protein or extracellular domain of the membrane proteins have the oxidizing environment, that help these particular bonds to be formed. So the S have like in the oxidizing environment, it possess some negative charge that needs to be shared with the other molecule. So this would form a bond in the oxidizing environment. If we are reducing this, then it would be separated and it would form a SH bond. Okay. So all these places where the secretory proteins are there, lysosomal proteins are there, these are the oxidizing environment. And in this scenario, the proteins need to be stable. So disulfide bridges are formed in this case. Yeah, cysteine. These are formed between the cysteine residues. Okay, any doubts?
I thought they are hydrophobic, that's why they are free of water. And for reducing, we have to add water to add these proteins,
No, no,
secretory
no.
protein, lysosomal proteins.
Uh,
And they will be accordingly oxidizing or reducing environment.
Yeah, because here we are talking mainly about the oxidizing and reducing environments. The disulfide bridges are linked to that only. Okay, it is not related to the hydrophobicity of the protein. Even in bacteria, whenever we see the periplasmic proteins that are present there, they have the disulfide bridges because of the oxidizing nature of the periplasmic space.
So, what is pH in mitochondria and
pH in
prokaryote?
mitochondria is less than the cytosolic pH because there also the hydrogen it varies. Okay, because if you are talking about mitochondria, so these are the Christie. Here it's low pH in the intermembrane space. Okay, because all the hydrogen ions are accumulated here, and when the hydrogen ion comes to the inner core of the mitochondria for the ATP production, so then the pH gradient is maintained in that case. So this is a low pH region, and this is the cytosolic pH, that is around maybe 6.8. That's what I remember.
Because I thought it is nearly same, 7.2 to 8 means.
Yeah,
uh,
the inner part is nearly the same. Okay, it's at the high pH like the cytoplasm, but the intermembrane space between the cytoplasm is maintained at low pH, a relatively low pH, because the ATP production comes into play here. Okay, is it fine?
And same pH is with endoplasmic reticulum, no?
No,
7.2.
yeah, endoplasmic reticulum is a bit what I say. In case of the
Mhm.
endoplasmic
Mhm.
reticulum, the disulfide bridges are formed there itself. Okay, so uh, it's like I think it would be similar because it's in direct contact with the cytoplasm. Okay, so endoplasmic reticulum would be similar.
Because they have neutral pH. It is in comparison with the cytosol or actual pH scale from 1 to 14. That's question.
Hmm.
And what does this neutral pH has to do with this secretion?
Okay, so neutral pH in that sense is endoplasmic reticulum have various chaperones also in their scenario. Okay, so the endoplasmic reticulum have it will not be similar to cytoplasm, it will be a bit less. That's what I remember. Okay, because all the protein folding starts from the endoplasmic reticulum, and the protein folding and the this thing disulfide bridges formation, it requires relatively low pH as compared to the cytosol. Okay, so endoplasmic reticulum has somewhat low pH as compared to the cytosol. That's what the case can be. because all the secretory proteins and this lysosomal proteins follow this pathway from the endoplasmic reticulum to Golgi, then to vacuoles and the outside. Okay, so let's stop the class for today. Okay, so if you guys have any questions related to this particular presentation or this week, you can ask. Yes.
Sir, so can we say that we can't actually predict whether the cytosol is having oxidizing environment or reducing environment? It depends.
Yes, actually, cytosol doesn't have an oxidizing or a reducing environment because it's maintained at a normal pH. All the normal activities, translation, even some of the protein folding happens in the cytoplasm. So it is present at a neutral pH. It is not neither oxidizing nor reducing environment in the cytosol.
So we can say that that where the protein are synthesizing, particularly that environment will be oxidizing because their bonds are formed, just like in the case of disulfide bond.
Yeah, the protein synthesis happens in the cytoplasm, right? Although it is linked to endoplasmic reticulum, but protein folding happens in endoplasmic reticulum, right? So in the endoplasmic reticulum there is somewhat oxidizing environment maintained, so that because the secretory proteins are formed in the endoplasmic reticulum, not formed, folded. Okay, so there somewhat oxidizing environment is maintained.
Sir, like for the secretion of protein, does this environment affect, like whether the environment is oxidizing or reducing? Will it affect the process of secretion? Like the protein is synthesized. Now it just need to secrete and need to go to the destined place.
Hmm, hmm, hmm. Okay, so in that scenario, I don't think because the protein needs to be stable in that environment, right? For example, if it's a protein that is present on the membrane, so this is the cell, and here the protein needs to be present. Okay, so in that case, the protein needs to be stable in this environment, right? So if the endoplasmic reticulum is making the protein and then delivering through Golgi and vacuoles, so it is maintaining that environment in which it remains stable.
So
Okay,
please,
sir.
can you explain with an example like mast cell or
Sorry, can you repeat?
the secretory protein like mast cell or anything other secretory protein or lysosome? Any example? Can
Okay,
explain.
any example? Okay, so let's take the example of immunoglobulin molecules. Okay, I hope you know antibody molecules, right? So antibody also have a disulfide bond. Okay, or do you know antigen presentation? If you guys have studied immune system,
Yes, sir.
okay. So, but leave it. That will be a complex procedure. Okay. So you know about the antibody, right? So this is the both the chains, and those are linked by the disulfide bridges, right? And so what will happen? For example, if we take the scenario, this is the nucleus. Okay. Now the endoplasmic reticulum is attached here. The protein is formed on the ribosome, right? Now when the ribosome is forming the protein, it moves to this particular compartment. So the ribosome is attached to the ER on the surface, and the protein molecule is produced towards the inside. So it directly moves to the core of the endoplasmic reticulum. Now what will happen here? different chaperones come into play. Like, for example, the two basic chaperones that you would have studied is carnexin and reticule, right? So now what happens? These two chaperones help in the folding of the protein. So here only they start folding the protein. Okay. Now here, because of the low pH, what will happen? The two chains that needs to be having the disulfide bridges also are formed here. Fine. Now, if we move from here to now, this vesicle will be formed and it will move to Golgi. Right now, through different phases of Golgi, that is from the trans to the cis phase. Now, sorry, cis to the trans phase, it will move, and different other modification like glycosylation and other different modification happens in Golgi. Now from the Golgi, it will protrude out in a vesicular format. Now here, the antibody molecules will be arranged in such a way that I'll make it big. So this is a vesicle, right? An antibody molecule has a membrane domain also. So this would be arranged in such a way that this is the membrane domain, and then it would be projecting inwards. This is the heavy chain. This is the heavy chain, and the light chain would be attached like this, right? So likewise, this is a immunoglobulin molecule that is being formed from the this procedure after protein folding. Okay. Now, after this, this vesicle basically merges into the cell membrane. Okay. Sorry. So, after merging into the cell membrane, it will be like this. So, this is the membrane domain. This is the heavy chain, followed by the light chain. So, now the basic picture is clear, or any more questions on this? Right. Now, the second component, if this, because this antibody molecule is not every time the membrane bound, it's a secretory protein. Okay, the actual antibody that is directly bound to the antigen or that helps in the immune response is secretory in nature. So in that case, what will happen? Only the membrane domain remains till it is the sorry, the membrane domain attaches only when it's in the Golgi. Okay. After it is removed from the Golgi, the membrane domain is removed, and only the antibody, the mature antibody molecule remains in the vesicle. Okay. Now here, from here, it attaches to the cell membrane and it is released into the outside solution. And there it is basically bound with different proteins that helps in maintaining its structure at different particular environment that is maintained outside the cell. Okay, is it fine now? No. Okay, so shall we close the session for today? Okay, thank you guys for joining. If you guys also have any doubt, go ahead and we'll discuss in the next lecture also. Yeah. Okay, okay, okay. Thank you guys for joining.